What is an embryo? An embryo refers to the early developmental stage of eukaryotic organism resulting from the fertilization of an egg delivered from a female and sperm delivered from a male as a method sexual reproduction. In animal, the initial diploid cell that result from the diffusion of the egg and the sperm contain half genetic information, which is DNA from each of the parental cell, and is termed a zygote. As the zygote begins to divide by mitosis, the early multicellular organism is called an embryo. What is embryo splitting? Embryo splitting involves splitting the multicellular embryo at an early stage of development to generate twins. This type of cloning occurs naturally and has also been performed in the laboratory. Example. One of the examples of animals that have been undergone embryo splitting is sheep. Stan Willardson demonstrated that twins could be produced in sheep and cattle after splitting cleavage stage embryos and transfer of the dummy embryos into recipients. These studies demonstrate that triplets and even quadruplets could be obtained, even if at the lower frequencies due to the loss of cellular mass.
As mentioned, embryo splitting is a biotechnology method used in breeding animals, and one of the most important purposes of this technology is to create or generate a monozygotic twins with desirable genetic characters and multiples it successfully. This will continue to become an economic factor in animal farm breeding. Embryo splitting allows it to obtain ideally several embryos with identical genetic features from one embryo. Since monozygotic twins are genetically homologous, it is possible to obtain an individual with excellent traits that we interbreed, and this will give us advantages. Year by year, embryo splitting has been extensively used in veterinary medicine and breeding as purpose to maintain high quality and healthy livestock that demand. With this purpose, embryo splitting technique is still relevant and used in practical application for reproductive management.
As we know about the purpose of embryo splitting, now we go through to the how embryo splitting was done in animal. First of all, the embryo splitting is the way of how the animal was cloning. Embryo splitting is done by reproductive cloning, involve the plantation of a clone embryo into a real or an artificial uterus. After that, reproductive cloning experiments were performed through the process of embryo splitting, in which a single early stage 2 cell of embryo is manually divided into 2 individual cells and then grows as 2 identical embryos. So, I will talk about how the embryo splitting was done. Firstly, as embryo is split, that form 2 half embryos. The embryos then are transferred to an unrelated surrogate mother, and then the pregnancy is monitored by ultrasound and the animal will give birth to identical twins. To give more information about the embryo splitting, we can include the example of Dolly the sheep, who was generated through the process of somatic cell nuclear transfer, or called as CNT. Firstly, a donor cell will taken from a sheep udder and an egg cell is taken from an adult female. So, the process entails the removal of the entire nucleus from a somatic cell of an organism and followed by the insertion of the nucleus into an egg cell that has had its own nucleus is called enucleation. After that, once the somatic nucleus is inside the egg is stimulated with a mild electrical current and will start dividing. A clone embryo, which is an embryo of an identical twin of the original organism, is created and the clone embryo transferred to a place in the uterus of surrogate mother, and the embryo will develop into normally lamb, which is dolly. The SCNT process has undergone significant refinement, and the procedures have been developed to prevent damage to eggs during nuclear extraction and somatic cell nuclear insertion.
There is a few advantages of embryo splitting. First, high success rate for obtaining twins or multiple identical offspring. Second, embryo splitting should increase the likelihood for obtaining a pregnancy since more embryos could be made available for intrauterine transfer. Next, reduce the number of invasive procedures necessary for egg retrieval and the necessity for hormonal stimulants to generate multiple eggs. Next, the disadvantages of embryo splitting. First, not all embryos are likely to survive. Next, the clones are made from embryonic material, so it is not possible to clone an adult animal from which its cells have already differentiated.